Welcome back to Day 9 of NCERT Biology 365. Today, we study sections 8.5.9 through 8.5.11, starting with the centrosome. Open your textbooks to section 8.5.9. The centrosome contains two centrioles lying perpendicular to each other, organized like a cartwheel. Unlike cilia or flagella, which have doublets, the centriole is made of 9 peripheral triplets of tubulin protein. Its central region, called the hub, contains no microtubules, creating a 9+0 array. This difference is a prime target for statement-based questions. Now let's look at the nucleus. First described by Robert Brown in 1831, its internal nucleoprotein material was later stained with basic dyes and named chromatin by Fleming. While most eukaryotic cells have a nucleus, NCERT highlights two major exceptions that lack one: mature mammalian red blood cells and the sieve tube cells of vascular plants. Do not make the mistake of assuming all living eukaryotic cells must have an active nucleus. The nuclear envelope is a double membrane separated by a 10 to 50 nanometer perinuclear space, which acts as a barrier. It contains nuclear pores, formed by the fusion of its two membranes to allow passage of RNA and proteins. Inside the nucleoplasm lies the nucleolus. Note this detail. The nucleolus is not membrane-bound, meaning its contents are continuous with the rest of the nucleoplasm. It is the active factory for ribosomal RNA, or rRNA, synthesis. Eukaryotic DNA is highly organized. During the interphase stage, the nucleus contains chromatin, which is composed of DNA, positively charged basic proteins called histones, non-histone proteins, and RNA. Think about this scale. A single human diploid cell contains about 2 meters of DNA packed tightly into its microscopic nucleus, distributed across 46 chromosomes. This packaging relies on histone proteins, which we will examine in detail in Class 12. Every chromosome has a primary constriction called the centromere, which has disc-shaped kinetochores on its sides for spindle fiber attachment during division. Based on centromere position, we classify chromosomes into 4 types. Metacentric has a centromere in the middle. Sub-metacentric has it slightly off-center, creating a shorter P-arm and a longer Q-arm. Acrocentric has it close to the end, leaving one extremely short arm. Telocentric has a terminal centromere. Some chromosomes also have a non-staining secondary constriction at a constant location, which looks like a small fragment called a satellite. Finally, section 8.5.11 notes that plant and animal cells contain microbodies, which are minute membrane-bound vesicles packed with various enzymes. This completes our page-by-page study of Chapter 8. Now head down to the description, click the link, and complete the Day 9 CBT practice quiz. Also, don't miss our podcast form of Day 9. Tomorrow, on Day 10, we will review the chapter summary, analyze NCERT exemplar questions, and take our 45-question final mock test. Click to subscribe and keep your daily streak going.